Professor Chalfie traveled to England to work with Professor Brenner at the MRC Laboratory of Molecular Biology in Cambridge, where he actually took upon a project which was started by Dr. John Sulston on the genes involved in mechanosensation. Dr. Tulay Hazelrigg, a colleague, and also his wife, was among the first to attach GFP to other proteins, demonstrating it as a marker. And we know that the protein GFP since then has become a fundamental tool of cell biology, developmental biology, genetics and neurobiology, and many other studies related to biological sciences to investigate diseases and several processes in model organisms. He also received Golden Goose Award for this work in 2012. Currently at Columbia University, his lab is engaged in investigating various aspects of nerve cell development and function using nematode C. elegans as a model organism. They work on neuronal degeneration, microtubule structure and function, and channel structures and functions as well. They are currently studying the genes that act to transduce touch. A fellow of American Academy of Arts and Sciences, he's a member of National Academy of Sciences, National Academy of Engineering, and National Academy of Medicines, collectively called as National Academies in the USA. He's there as a chair of the Committee of Human Rights of National Academies. I can confidently say, as it also appeared during our interaction, Professor Chalfie is a vocal advocate for basic research and training in the sciences. With this very brief introduction, I now invite Professor Chalfie to deliver his lecture on Guarantor Transcription Factor in Cell Differentiation. Professor Chalfie.
Thank you. Is this on now? You can hear it? Good. Because I tend to roam a little bit away from the podium. Thank you. I really appreciate the introductions and the opportunity of being here. It has been a sort of hectic couple of days, but one of the things I'm really excited about is some of the other talks I've given have been about the past. This is what we're currently excited about in the lab, so I'm really thrilled. I personally think that you can get any Nobel Laureate in the world to talk about his or her current research, and they'll come running, because usually people ask them to talk about their Nobel work, which is decades old and maybe not as interesting. But this is a chance I get to talk about general work. But before I do that, I have a little advertisement that I have put in the beginning of all of my talks that I'm going to try to convince you to do something. It's something we do in our lab. I find it very useful, and I would like to have as many people all over the world doing it in their labs, if at all possible. And this started off because I was invited to a meeting chaired by these four people of something called ASAP Bio. ASAP, Accelerating Science and Publication in Biology. And what these four people, and actually about 100 or 200 people at the meeting, tried to do is to talk each other into, it's very easy to do that, to using a repository for preprints, that is, papers before they're published. And so to use what usually is referred to as a preprint archive. Now, the people in physics have been doing this for over 25 years. When they write a paper, they do send it to journals, but they also put it on a preprint archive so that every single person in the world can see it. And this meeting about a year and a half ago, 2 years ago, was to try to see if we could promote this in biology. And I thoroughly like the idea. Some advantages of using biology preprints. First, it speeds up science. Instead of, well, the characteristic thing that happens in my lab is we write a paper, we're ready for the world to see it. It gets sent to a journal. They spend 2 weeks trying to decide, is this paper worthy of our journal? Should we send it for review? And if we're lucky, they send it for review. The reviews take usually a month or two, sometimes even longer. They come back, they never come back saying, oh, this is wonderful. They always come back with this laundry list of things that we have to do. We spend a couple of months, we do all of those, we send it back. Another month goes by, and then they reject the paper, and we do it all over again. All of this time, the only people that have looked at the article are the two reviewers, or two or three reviewers, the journal people. The rest of the world hasn't seen it. We're done with the work. We want the world to see it. By putting it on a preprint server, and the one we use and many people use is the one at Cold Spring Harbor. It's a non-profit one called Borrow Archive, which the website is there. People see it from day one. The second thing about it is that instead of it going to a journal, eventually being rejected, maybe going to a couple journals, and the last journal, the one that actually publishes it, you get a timestamp of when the paper was submitted. But the paper might have been submitted a half a year, maybe a year before that time. Putting it on the archive gives that original timestamp. So there's no argument about who had the idea, who promoted different things. Yep, okay, I'll try to use the pointer here, although it's pretty simple, I think, to see. Anyway, accurate timestamp. I don't think that's important. Here's the reason I think everyone should do this. It makes the research universally available. It's free. Anyone that has access to the web can look at the article. They don't have to be in a rich country. They don't have to be at a rich university. They don't have to be in a university. It could be high school students. They could be interested reporters, anybody that would be interested in it. It's universally available for free. Next thing is that at these meetings we have talked to various funding agencies, other organizations that are publishers, and they've all agreed to do this. And so it used to be, for example, in National Institutes of Health, if you wrote a grant and they said, what has been your progress? The only thing they would allow is a paper that was actually published or in press. Now, they've changed the rules entirely. In press, published, or in an archive. So they get to see it for hiring, for promotions, all of these things now. The growing trend is to accept papers in archives. So everybody's work, the total sum of the work is what's considered for all these various things. I think I have one more thing here. And this is what I'm really getting at, that you have a potential that a much larger audience can help improve the manuscript. And this is what I really want to push, because this started out as an idea I heard from Francis Collins. He's the head of the National Institutes of Health in the United States. And he was telling me one day, we were at a dinner together, he said that he has a journal club in his lab, and they took a paper from the archive to review. And I thought this was brilliant. So I have stolen the idea. I want you to steal it as well and use it. So the idea is, and this has to do with the fact that my lab is sort of small. People can't really give a lab meeting on their work every week, so we alternate lab meetings talking about the research and a lab meeting in which someone picks a paper from the archive and everybody reads it. Technicians, graduate students, undergraduates, postdocs, myself, everybody in the lab reads it, and we have about an hour-and-a-half discussion about the paper. After we've had this discussion, as I said, it must come from a preprint server, so they're the latest material. After the hour-and-a-half discussion, the person presenting the paper gets all the comments that we have had about it and writes up a summary and sends all that information to the authors. And we say, here, we're trying to be helpful. We have actually looked at your paper. Here are our thoughts about this. If you find them useful, you might want to incorporate some of these ideas in your paper. Or we didn't find this part as understandable as I think you think it is. And we send all the information to the authors, and we also tell the authors about this journal club so they can do it as well. And that they can set up something similar. I also, right now, our standard procedure in the lab is when we have a paper we want to submit for publication, we send it to the journal, and simultaneously we send it to the archive. And when we do that, I then send an email to everyone I imagine might be interested in the results that I know of, and tell them the paper's on the archive, here's the link, and here is what we do. We have a journal club. We don't do this publicly. We don't publish our comments about a paper. We don't put them in a comment section. We simply send them to the authors, hoping that we can be helpful. And so the advantages of this are, first, the lab members are reading the latest material in their field. They're not waiting for something that might have been new a year or more ago. This is the latest information. We always want our students to be participants in journal clubs because it gives them skills in reviewing. But those skills, it's sort of an empty exercise if you take a paper that's already published. Those authors are done with it. There's no usefulness in the review other than to the students. Here it's useful to the students and to the authors. And so the students and other people in the lab realize that they are actually, while they're learning, they're helping other people. They're being useful in what they're doing. And that's a wonderful advantage. Obviously, the authors get additional reviews. And in those reviews, we don't say, we think this should go into this journal, or we don't think this is good enough for that journal. We don't care about that. We only care about the science and talk about the science. And I have to say, the reaction has been very positive. I have another ulterior motive of why I'd like everyone in the world to do this. I think one of the horrible things that's cropped up in the world of science these days are these journals that are usually referred to as predatory journals. We will publish your paper, no matter what it is, because you're going to pay us $2,000 or $3,000. And so they get money, you get seemingly a publication. But if everyone is going to be considering papers put on the archive as useful, and it costs $0 to put it on the archive, why would anyone waste their time paying somebody in one of these predatory journals? And so my idea is that if enough people do this, we can get rid of the predatory journals and at the same time really promote sharing and building a sense of community, because we're all in this together. This is not me against you, it's all of us trying to make all of our science as good as it can be. So I really want you to consider doing this, to set these up in your own labs and see how it goes. See if it's not as useful to you as it has been for us. So now I'll give the real talk. So for many years, starting in 1977 in fact, I have been working on the nematodes Cenorhabditis elegans and the cells here that are drawn in blue. They're very simple shaped cells. They're nerve cells, and they're the cells that allow this animal to sense touch. Now, the animal as an adult is only 1 millimeter long, so sensing touch in such a tiny animal has required a very sophisticated piece of equipment. We take an eyebrow hair, we glue it to a toothpick. That's the equipment. Under the microscope, if we stroke the animal in the head, it goes backwards. If we stroke it in the tail, it goes forward. If these cells are missing, it doesn't move. So it's very easy to collect mutants that are insensitive to touch. You just tickle them with the eyebrow hair after you've treated them with a chemical, for example, to make mutations, and look for animals that don't move. There's a slight problem with this. Animals that are dead don't move either. So you need to we have another way of making the animals move forward that doesn't depend on these cells. So if they don't respond to gentle touch, but they do respond to a harsh touch stimulus, then they are defective in these cells. And over the years, we've collected quite a number of mutations, over 500, identified the genes that were defective in these, and I'm going to talk about a subset of the genes. I'm going to talk about the ones needed for the development, because a way to make an animal touch insensitive, there's two general ways. One way is don't make the sensing cells, and that allows us to ask questions about development. I'm going to spend my time talking about that. But the other thing is a cell could be made, but it just doesn't work. It doesn't function. And that has led us not into studying the development of specific types of nerve cells, but rather what is the molecular basis of mechanical sensing. Scientists have known for 130 years, something like that the molecule needed for us to detect light, to have vision, is the molecule rhodopsin. And we've known for about 40 years the molecules, the receptors that allow us to detect chemicals, whether they be odors or tastes or internally neurotransmitters or hormones. We know the chemical receptors for that. So we know chemical sensing to a pretty good degree. But we have lots of senses that are mechanically driven. Hearing, touch, stretch of our muscles, detection of blood pressure, all sorts of things. And we're basically still pretty clueless as to how these work at a molecular level. And so being able to get mutants in which the cells are made but don't work, one of the hopes we had is that we would actually be able to find the sensor, usually called the transducer. And I'm not going to talk about that work, but in 2005 we published a paper actually showing that we had the first sensor from any nerve cell and that would detect mechanical signals. But I want to talk about the development and maybe present a couple of developmental questions first. So what are some of the questions? Of just looking at these 6 cells out of the total of 302 nerve cells in the animal, one question immediately comes up is, how do you get this particular type of nerve cell? These cells have very distinguishing characteristics. What allows them and no other cells to have that characteristic? And I'll talk a little bit about that. Another one is it enough to just flip the switch and say, okay, now you're going to be this type of cell? Or do you have to keep the switch on? Do you have to maintain it? And the answer is you do have to maintain the development. It's not a one-time switch, but a switch that then has to be held in place. How does that come about? Another question that comes up is, how do these very different cells actually take on this particular fate? Now, all 6 of these cells, you can say, well, an easy way is there's a precursor and it divides and you just get these 6 cells and they migrate to where they're supposed to go. That would be a clonal way of doing it. That's not what happens here. Each one of these cells comes about almost completely independently from all the others. And if you ask, what's the one common cell that eventually gives rise to all 6, you have to go back to the very, very early embryo. I think the 24-cell stage, or even earlier than the 24-cell stage embryo. So how is it that a cell over here and a cell over there and a cell over there suddenly have exactly those same characteristics? And we have no idea what the answer to that question is. Another question. If we look at these cells, there's some very subtle differences. These 2 cells, ALM on the left and ALM on the right, are cells that have a cell body and a neurite that goes up towards the tip of the nose. They are monopolar cells. They have only one process. Whereas the cells in the back, they have cell bodies that have this anteriorly directed neurite, but they also have another neurite that goes in the back. So they're bipolar. Why is it that some cells are monopolar, the other cells are bipolar? There's other slight differences. These cells are closer to the dorsal surface than these cells, which are closer to the ventral surface. And they also make different synapses. So how do all of these differences come about? And the final question, and the one I'm going to spend most of the time talking about, is a very strange thing, or at least struck us as strange. And that is, there are two of these ALM cells, one on each side. Every single animal has two cells. The animals in their development don't seem to make mistakes, or they do it so rarely we've never seen it. Now, those of you that have ever been in a chemistry lab know that reactions don't work all the time. There's a lot of variability. So we would expect, since it's really a chemical reaction that starts these cells down the road to become a certain type, we would expect it to fail some of the time. And then we get animals that would have only one cell, not always two. But we always do. And that robustness, that surety that we have that every animal will always have those two cells, really intrigued us. And that's what I'm going to spend most of my time talking about. How can cell types be generated so robustly? Now, the engineers have an answer for this. They know how to get a chemical reaction or any event to happen whenever you want it to happen. And that is, have more than one thing. Make things redundant. And in biology, that happens as well. So redundancy can make sure that a gene is expressed. And Mike Levine's group wrote this nice review in 2012 in which they said, look, we've already found examples of redundancy, and we're going to tell you them. And so if we look at this top line, the region that activates the expression is often called the enhancer region. As part of a basal promoter and then an enhancer. And so in the first case, if there happens to be a mutation that knocks out that enhancer so it can't, the transcription factor, TF, won't bind, then it can bind to another enhancer called the shadow enhancer, and development will proceed. Or there can be multiple binding sites within an enhancer such that if one binding site is missing, the transcription factor can bind to the other site. Or even if there's only one site and you have a transcription factor binding to it, you get rid of the transcription factor, there's a redundant transcription factor that can bind to the site so that it works. These are tried and true methods. In us, our nervous system, and is all mammals, the nervous system, as the organism develops, produces twice as many neurons as they need. And the ones that don't hook up correctly, they're killed. So that's redundancy as well. But there's a problem with this type of redundancy, and that problem is it works too well. It absolutely ensures that this is going to happen. So if you wanted to change something, you'd have to change all of the redundant elements. Everything had to be changed in order for it to change because it's designed to overcome possible defects and changes. So redundancy is great for doing what it does, but then it can't be modified. Okay, so as we were looking at the genes that we had discovered were needed for touch sensitivity, first set of experiments looked at two transcription factors, UNC86 and MEC3. And the simple question, or the simple answer here, is that what we found is that the UNC86 gene turned on the MEC3 gene. Now, once that was turned on, both of these came together as a dimer, and they turned on a whole bunch of genes downstream that made the cells the particular cells they were. And MEC3 was only made when the cell was generated. So the UNC86 MEC3 complex, that dimer, is what controlled the development of the cell. But it also had another function. And that is that once this dimer was made, it would come back and activate the production of more MEC3. So this is how maintenance takes place. You turn on something and then have it turn itself on and keep itself on in a positive feedback loop, so positive regulation. And we were exceptionally happy with this whole idea. This answered everything that we wanted about development. We had what was usually referred to as a terminal selector, and this turned on a battery of genes that made the cell, which are called differentiation genes. And when we did this work, culminating in like 1993, we were very happy with this because we figured we had figured out the development of the cell. And then a horrible thing happened. I had a good postdoc who did one more experiment. And that was Irini Topolito. Now, I always at this point I need to stop and talk about the code here. You're going to see citations, people's names in red. All of those people worked in my lab. Blue, these are collaborators. And people whose names are in black, I'm very envious of. I wish I had done what they did. So Irini was in the lab, and she was isolating RNA from the touch receptor neurons, and she found another transcription factor. That was the target of all of this. This was turning on another transcription factor. So she started working on it, and she got some very strange results. So for example, if we touch the animal 10 times, the wild type animal, you see each dot is an animal here. You see that the animals respond almost 10 times out of 10. But when we got rid of the ALR1 gene, we got this incredible range of touch sensitivity in the animal. And these are 3 independent mutations. Each one gets rid of the gene. So there's an incredible amount of variability, and it was this variability that intrigued us. And the reason for that is, first of all, we can rescue this by putting the gene back in, and we get back to this level, more or less. But we had other mutants that gave a partial touch insensitivity, but when we looked at those, they gave a small range, not this extensive range that we see here. And so we were very puzzled by this and wanted to think about this. And let's see. And it turned out that there was another group, Piali Sangupta's lab at Brandeis, that had also looked at this same gene and said, you know, in the cells we're looking at, the phenotype is really variable. It's all over the place. So that was intriguing. 2 different sets of cells, 2 different labs doing the experiment, a great amount of variability. So Irini set off to try to understand what was going on. And she had developed a very nice yeast system to look at gene activity and used the MEC3 gene, now coupled to LAC-Z from E. coli to make beta-galactosidase, which you could see as a red dye that you could measure. And you can see here, nothing here. This whole thing is the control. There's no real element. Nothing happens. But if you look here at the MEC3 upstream region, the MEC3 promoter, then what you see is that ALR1 does nothing. UNC86 MEC3, which should turn this on, does a fine job of turning this on. But when we add ALR1, the thing that did absolutely nothing, now we get about a doubling in the amount of transcription. If we use another promoter that is expressed in the cells but is expressed in all neurons, it's not specific, again, none of this stuff works on it. So this is saying that somehow ALR1 is doubling the amount of expression. And we wondered how that doubling would take place. So this is our theory of what we thought was happening. We know that if we were able to look at individual molecules of messenger RNA and count them in a cell, that if we look at a whole bunch of cells, there should be some distribution. So some cells would have very few, some cells would have more. We'd get this sort of normal distribution. And we imagine that one way to double the amount of material that would come out of this is if we just shifted this curve to the right, something we're calling enhancement, and we double the amount of activity. So this is what we expected. But you might be asking yourself, can you actually do this experiment? And fortunately, we were able to collaborate with Alexander von Oudenarden at MIT, and we were able to do this experiment. Now, the way this method, called single molecule in situ hybridization, works is you look at your messenger RNA, and you go send off a bunch of money to a company, and the company sends you back small probes, 20 nucleotides long, each with a fluorescent molecule attached to it. And each one of the 20 goes to a different part of the RNA, so you just decorate the whole RNA with about 60 different probes, each one with a fluorescent molecule, and that's enough to see in the light microscope, the fluorescence microscope, each dot is a single messenger RNA molecule. So you can count them and go through a Z series. So here you see the whole animal, and this particular gene is expressed in these touch cells. These touch cells, there's another set of cells up here that expresses them. We can simply count the dots. And when Irini did that, without ALR1, we got a distribution. Now remember, we want this distribution to head towards the right. But when we looked at that, what we got is we lost the bottom, and we didn't get really much higher up here. We just collapsed this to a much smaller region. In other words, we didn't have enhancement, but we had something we called refinement. And this doubled the amount of expression. But we never really got above what we had had here before. So how do we think about this? I think about this in a very simple-minded way. Actually, I probably think of everything in a very simple-minded way. But the idea is, if concentrations are the same, there are going to be three things that are going to affect transcription. So we have our dimer coming and binding to the DNA. So one thing that's going to be important is the on rate. Another thing that's going to be important is and that's usually diffusion. The off rate, which is going to depend on how well these are stuck here, so what the binding constant is, that's the off rate. And overall, we'll have some sort of equilibrium here. And then once these are bound, they'll have an intrinsic ability to turn the genes on. And so as all these things might vary, you would get this sort of distribution in the cell. But if now you had another component, and ALR1 actually binds right next to the UNC86 MEC3 site, now it could be doing a whole bunch of things. We'd think it a little unlikely that it's opening up the chromatin because this works without this, so we don't think the on rate is affected. But by providing another binding surface right here, it could keep this from coming off, and so the off rate would be greatly reduced. Or it might actually bring in an additional activation probability that would help things here. But we couldn't get any more activation than having this at its maximal amount. And we'd be able to get this refinement. We think the variability that we're seeing occurs in this region here. That there's just isn't enough MEC3 for the cells to develop, and they develop to a various extent. So an intriguing thing. Transcription factor that doesn't turn on transcription, but is needed for another transcription factor to work optimally. And so we've now added ALR1 to our story here, and we're calling this a guarantor because it guarantees this maximal expression. And we were quite intrigued by this. And then we forgot about it for a couple years. I had a new graduate student come to the lab, and he was very interested in that question about the touch cells being different in the head and the tail. And so he wanted to study that. Now, The minute anyone in developmental biology starts talking about differences between the anterior and the posterior of an organism, the thing that most of us think about is the work of Ed Lewis. He won his Nobel Prize in 1995, shared the prize, in which he was able to look at development of Drosophila, and he was interested in some fascinating mutants called the homeotic mutants. And these homeotic mutants, if they were defective, sometimes instead of having a normal fly with just a pair of wings and then these small little bits that you can hardly see here called halteres, now a part of the thorax was changed into the more anterior part, and now you got two pairs of wings, turning the flies into dragonflies, if you will. And so this is a profound developmental change where one part of the animal now develops as another part of the animal. And a lot of genes were known that could do this. Some of the more famous ones, there was one called antennapedia, where instead of having an antenna grow out of the head of the animal, it was a leg. So it had been completely changed. And these were known as important developmental genes. And one of the most astonishing thing about these genes is that they lined up on the chromosome. So that the first one, labial, actually was expressed in the head. Then this is called proboscipedia, deformed, and so on down here is antennapedia. Normally it's expressed down here, but in those mutants it was inadvertently expressed up in the head. You get a leg instead of an antenna. So this was known for this regional specification. And the remarkable thing is that these same genes are found in vertebrates, in us. And they seem to be needed for the development of the different parts of the nervous system. So the same regional specification, very exciting. However, as people have studied these genes more and more, they've discovered that these genes have two functions. One function is this regional specification, anterior going to the posterior. But the other part is that no matter where they're expressed, they're needed for cells to differentiate. So for example, there are airway cells, tracheal cells, in the fruit fly. They are in every part of the fruit fly, but these genes are responsible for making sure they do get made. So there's not a regional difference. It's the same type of cell. It's just being made in all the different places. And as people, Tom Jessel's group among others, have looked in the mammalian nervous system, they have found that there too, these genes, which are usually referred to as Hox genes, are also needed for this general cell differentiation. They're needed for that. So when Xiaogu Zhang started in the lab, he wanted to study originally why the ALM cells were different from the PLM cells. And so he did a series of experiments, many with an undergraduate, Felix Tsien, who went on to be the valedictorian of his year. And they looked at the Hox proteins in worms. Now, if you have a photographic memory, you may say, wait a minute, in flies, the red box was before the yellow one. There is a rearrangement here, which makes it a little strange, but they only have 6. So it's a really simplified system. And in fact, of these 6, what Xiaogu and Felix found is that only one, EGL5, is expressed in the tail. The others you can ignore, except for say 13, which is this red one, and that is expressed in the head and in the tail. And that sort of followed us up a little bit. And then we realized that this transcription factor needs another transcription factor, a co-transcription factor, which is called usually PBX in flies, and in worms it's called SAY20. And SAY20 is only expressed in the head. So this can only work in these cells. It can't work back here because this is not back here. So we have one that's in the back and one set that's in the front. And they wanted to know what happened. So they took an animal that had a very strong promoter to drive red fluorescent protein. And in a newly hatched animal, they could see, the only 4 cells that are there, they could see these cell bodies very nicely expressing RFP. They are differentiated cells. And they asked, okay, if we put our mutants in this, what do we get? And so this is one result. Here is either a SAE13 or a SAE20 mutant. You still get the nice expression in the tail, but the expression in the head is gone completely. Now I should tell you, I don't have pictures of this, the cells are still there. So the cells have been made, they just do not differentiate as this particular type of cell. So the cell is made, it's there, it doesn't express this downstream function gene. In fact, they looked at many function genes, and the story is always the same. They don't see it. They looked in the tail. Now they got rid of EGL5. And when they get rid of EGL5, now you see the expression in the head, nothing in the tail. So these transcription factors are very definitely needed for the differentiation of these cells. But I've left one important fact out in describing their work. This might lead you, if this happened all the time, it would lead you to think that these are important transcription factors for the development of these cells. Without them, the cells don't differentiate. But that's not true, because these beautiful phenotypes only occur 40% of the time. The rest of the time, the cells are perfect. They don't need these genes. It just doesn't work 40% of the time. 60% of the time, for example, up here in the head, if we had a mutant where these were gone, but the cell was still made, the animals were touch sensitive in the head. So the cells not only have differentiated, they've differentiated all the way to the point of being completely functional without having to have these genes at all. So it only works 40% of the time. And that's when we started to think about ALR1. So we started to do a couple of other experiments, similar to the ones I've already showed you. So for example, we looked at the messenger RNA. So here's the wild type distribution of MEC3. And in SAE13, we get this bimodal distribution. Some of the cells are absolutely perfect. SAE20, same thing. Some of the cells are absolutely perfect, same sort of distribution. But then we have this population here in which it's really not working at all. And it will not come as a big surprise, I hope, to you to find out that that part is 40% of the animals that we look at, 40% of the cells that we look at. So it's consistent with what we saw with the differentiation. If everything goes well, we're fine. If it doesn't, nothing. They also did this with EGL5, and the same thing happened. Again, 40% didn't differentiate and express this gene, and didn't keep MEC3 going. The rest were just perfect. So they did some more detailed experiments. We have a very small, minimal promoter for MEC3, so it's only 392 base pairs, driving red fluorescent protein. The blue boxes are the ones that bind UNC86 MEC3. The orange boxes are the ones that bind the HOX proteins and their cofactors. And I can tell you that UM1 is unimportant, HP2 is unimportant, but these are very important. And so with this minimal promoter, they were able to show that they could get expression in all of the ALM cells, all of the PLM cells, but if they mutated this site, now they only got about 60. Again, this 40-60 split. Now you might say, well, wait a minute, this is pretty close to that. How do you know that when you interfered with this site, you didn't interfere with that site and it didn't work? Well, we were very fortunate. We have an internal control. You may remember that I said there were 6 touch cells, and now I've just been talking about 2 and 2. There's 2 other touch cells. And we don't know why, but they don't need Hox factors at all. So we can test whether this mutated version is any good by seeing what happens in those other cells. If this is compromised, those other cells won't express, but if it's able to work, then this will be fine. And the answer is perfect. ABM and PBM, the other two cells, are expressing even in the mutant. Again, suggesting that this mutation is really the important thing. So we also did the yeast to hybrid, the yeast system, where we took the minimal promoter, put it onto LAC-Z, looked at activity on its own without the MEC3 promoter, nothing. And we have this here, but we have basically nothing here on their own. So in other words, just like ALR1, they are not turning on gene expression on their own. They simply do not work. But yet, if we now add to this mix UNC86, which will turn on this promoter, then UNC86 alone gives a nice, healthy expression. If we add AGL5, we don't need a cofactor for that. We get about a doubling, just as we've had in other experiments. If we add the cofactor, it really doesn't do anything. There's not a big problem. What about the looking at the other things here? Now we have UNC86, but now we're looking at, say, 13. Not much, but when you add, say 13 and the PBX-like molecule, say 20, now we get this increase. What about with the mutant? No increases. So completely confirming the previous results that we had and suggesting that, in fact, this protein is binding here. So our overall view of what's happening in the cell is that initially UNC86 comes to lie on the promoter for the MEC3 gene and turns it on. But 40% of the time it fails. It can't get the expression. The Hox protein, which is there and not regulated by any of these things, serves as the guarantor, so now all the cells make this. Once they've made this, UNC86 can be made, MEC3 can be made, the dimer can be made, but that's sort of sloppy in and of itself too. But only when the second guarantor comes in do we now have this refinement, and we've narrowed this down. So in fact, in the differentiation, there's been two different instances where having this guarantor function has ensured that development occurs appropriately. Now, as if that weren't enough, I'm going to speculate like mad now. And one of the speculations that I want to present is those of you that know about the Hox genes and how wonderful they are and these beautiful phenotypes and everything, for the last 3 or 4 decades, people have been intrigued by these transcription factors, and they've asked themselves the question, what do these transcription factors turn on? What are the downstream things because they must be turning on all sorts of interesting things to cause these differences. And people have looked extensively. And it's an enormous problem because that experiment hasn't worked. It just hasn't worked. People have found a few downstream genes that are regulated, but not a lot. But these are major developmental regulators. We think that the guarantor idea changes the way we interpret those experiments and gives an explanation of why those genes actually couldn't be found, the downstream genes. And the reason for that is, first, that what we're seeing, what people were looking for, was a qualitative change. No gene expression, or gene expression, no gene expression, on or off. But we're not seeing that. We're seeing on 60% of the time to on 100% of the time. So we're looking at a quantitative difference. And people wouldn't have done their experiments to start off at 60% and then look for something that gave them 100. So we think that explains why people have not been able to find many downstream genes for this. And then there was a paper just about the time we were doing it, actually by a colleague of ours at our medical school at Columbia, Richard Mann. And in this paper, they were talking about one of the very few targets that is a target for Hox genes. It's called shaven baby. And as you can see here, well, they found two different types of binding sites for these Hox proteins. One, they said, for the specific regional expression, we find that we use very weak binding sites, but many of them. And those allow us to have this regional specific expression. And they said, but if you go in and look at what everyone else has said is a Hox binding site, these sites bind all the Hox proteins. They have no specification. They're high affinity sites, but they have no specifications. This was music to our ears because we would say those high affinity sites are Guarantor sites that would allow the differentiation no matter what Hox protein was binding, wherever it was within the animal. And so this gave us an idea. Why don't we look for high affinity Hox binding sites, and people have done experiments like this, and then see if they are actually, the genes that have them are actually using them as guarantors. And we've just really started on this project. Actually, it's a main project of an undergraduate, Kenneth Pham, in the lab. And Kenneth has already found a couple of candidate genes and has started to do some experiments, and the preliminary data suggests that there may actually be something about the guarantors acting in muscle differentiation. So not nerve differentiation, but it's still very early, so you should ignore that result, and we'll see what happens later. The other speculation I want to put here is that the guarantors address a question I raised at the beginning of the talk. And that question was, the problem with redundancy is it's hard to change it. But the guarantors provide a single component means of getting robustness. It's not multiple components. And so one could fantasize and say, well, wait a minute. Could this be regulated? So differentiation would go down one path one time and down a different path another time, maybe by phosphorylating or acetylating or otherwise modifying the Hox proteins so they either work or don't work as guarantors. And so we're starting to look into this possibility as a way of looking at the regulation of these components as a possible way of having interesting switches in development because these things are guaranteeing robust things. Maybe in some cells, you get 100%, or some organisms, you get 100% of one cell. Other times it's 50-50 or 60-40. And that would be a possibility. So I'm not sure how much time I have. I don't see a clock here. But I'm going to talk a little bit more because they also went to look to see about the regional specification. This time, Chagu worked with another undergraduate, Marguerite Diaz-Quadras, and she also was the valedictorian. So he's the best trainer of anybody I've ever seen in my life. He's just an unbelievable mentor. And they've all been great in the lab. And they wanted to know about the regional specificity. What about this cell monopolar, this one bipolar, this one more up, this one more down? And what happened, the reasoning was maybe one of these cells is the fundamental, the ground state, and genes change it from the ground state. So one model is there is a ground state that the posterior cells have, and these genes change it to the anterior fate. Another way, of course, is the opposite, that there's a ground state that these cells are already in, and they're changed to this cell, or there's some strange ground state and both things change it. So when they did the experiment, what they found was, in fact, that's the answer. That the ALM cells are in the ground state and they're changed by EGL5 in the PLM cell. to have these different characteristics. And the way they looked at this was first to get rid of the gene. And when they get rid of the gene, of course, it's not expressed in the head, so the head cell is perfect. This cell, instead of being bipolar, is a monopolar cell in about 90% of the time. In addition, sometimes this process comes up and runs in this track, where it never does before, because it's always in this lower track. But then they did the really important experiment, which was, let's put EGL5, which is normally only expressed here, and if that's really the control, let's put it up here, see what happens. And this is what they got. 40, 30, 20, 90% of the time, the cell became bipolar. Just as 90% of the time it failed to be bipolar up here. And as you can see, in several of the cases, there's processes here, and in this 20% of the animals, they were completely on the same line as this other cell. So we have a way of distinguishing these cells, again, the regional specificity by the Hox protein. So overall, what we've had, and this is going to summarize a lot of work, we have UNC86 and MEC3 causing a heterodimer that turns on a whole series of genes in both cells. Now, in order for it to work, there has to be this guarantor function to make sure it works, and it does, and also ALR1. But EGL5 also turns off some genes, like MIR84, making them seem like they're only in the ALM cell. But they could be actually expressed in both. So it's turning off some genes to give ALM-specific genes. And it's also turning on a bunch of genes just in the posterior cell. Now, we don't know how it's turning this on. Is it perhaps working as a guarantor of some other transcription factor? We just haven't gotten to that point yet. We don't know what this question mark component is. But we also know that EGL5 turns off a couple of other genes, specifically this cofactor. Remember, the cofactor had to be there for SAE13, and SAE13 was actually in both cells. Well, SAE20 is turned off by EGL5. Another cofactor is also turned off. So it's turning on some genes, it's turning off some genes. We have no idea how it's doing it. This is work for the future, and we're very excited about what this could be. And let me summarize overall what I think I've told you. We have a way of ensuring cell fate. Initially, the Hox proteins are making sure UNC86 turns on MEC3 in 100% of the cells. And then after that, ALR1 is making sure that the maintenance kicks in, and so we can maintain the differentiation and the optimal differentiation of these cells. At the same time, the Hox proteins are important for what's usually called posterior induction. For subtype specificity, we have a default state that doesn't require the Hox proteins, and a cell that's changed by EGL5 changing its features. And we've been able to find one of the components that it does for this. There is a membrane protein called RFIP1. If RFIP1 is missing, these cells are in the right place down here, but they're monopolar. If we take RFIP1 even without EGL5 and put RFIP1 in this cell, bipolar. So the bipolarity seems to be due to this final cell. Now I want to say one final thing. And this is the Balu slide now, because he suggested that I add this here. We don't know where this research is going to go from here. We expect that guarantors are going to be very generally applied, and we start to think about, would regulation of guarantors actually be interesting in all sorts of aspects, maybe even of human disease, maybe even of cancer, where these might change cell fate or something by changing it. No idea, a speculation. But this was a very different way of looking at these transcription factors, which is part of the reason we're excited about it. And it comes from basic research. that without doing this work of looking at this incredibly stupid, silly question, how does a touch cell in a worm develop, we wouldn't have made these discoveries about proteins, genes that are actually universal. And I have to say, over the years, it's often been like this. We keep discovering things by working in worms that then have applications and implications to human health and human disease. But everything has to start with basic research. And so I'm going to leave you with my favorite quote about basic research. I'm not saying that applications are not important. It has to go hand in hand. But you need the foundation of basic research in order to do the applications. And so in 1969, there was a wonderful congressional committee that was set up, and its sole purpose was to look into the possibility of this incredibly large particle accelerator that some scientists wanted to build, and was built in Batavia, Illinois. It became the Fermi accelerator. But in 1969, people in the Congress were a little unsure if they should spend so much money on what was really a science fair project. An extended science fair project, but nonetheless, a pure research project. Now, also 1969 was the time of the Vietnam War in the United States. There was a lot of feelings that were expressed, sometimes violently. People were very concerned about national security. They were very concerned about defense. And so when this congressional committee asked Professor Robert R. Wilson, who was the architect and designer and first head of this lab, if he would come in and talk to them, he came in, and one of the senators tried to make it an easy session for him, something that would win over everybody. He said, "Dr. Wilson, can you please tell us how many ways this particle accelerator will help national security?" And Wilson looked at him and said, "Senator, none." Not exactly the answer the senator wanted. So he tried it again. And a third time, maybe 4 times. Finally, he asked him, says, "Look, Dr. Wilson, will you just please tell the committee in how many ways, or in what respect, will this particle accelerator help the national defense?" At which point, Wilson said this wonderful thing. "It has only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with whether we are good painters, good sculptors, great poets. I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending." Thank you very much.
Well, I think last 1 hour he has taken us through the basic questions that he has raised in development using the C. elegans model, and the way they try to explain the differential gene expression by discovering or narrating their discoveries about Guarantor transcription factors, and several other questions. And more importantly, I'm happy that towards the end and also throughout the lecture, he has highlighted the importance of basic research. Thank you, Professor Martin, for the wonderful lecture. I'm sure you will be glad to answer a few questions. Now, the floor is open for questions, if any one of you want to ask something.
Okay. Grab the water here.
Here. So, okay, I just wanted to ask, I think, thank you, that was a great lecture, and also how one factor led to discovery of the other factor. So I was just working about the SAY20. So are you also looking at how that expression is regulated, and if when this EGL5 or was knocked out, that affected the expression of say 20 in the
In the
cell.
posterior
In
cell, yes,
the
it does.
posterior cell, it does.
Yes.
So, but now that our FIP1 precedes that, are you also looking that that also can may regulate?
We're looking. We like to look at everything. It depends on who happens to come to the lab and what they're excited
Okay.
about. But yes, there's
Yes.
all sorts of things here, and there's probably other things. There's an element of timing here that we have not really
Yes.
gone into. Taking that ALR1 story, if it was as simple as I said, something binding to the DNA and coming off and coming back on, then you might expect that without ALR1, things would change, right? It would come on, it would come off, and so we did that experiment. We looked at the animals and we said, how many times out of 10 do they respond? Some of them, as you saw, responded most of the time, some of them few times out of 10. And then we said, what happens the next day? And what happens the next day? And basically, it just stays steady. If it's low first, it's low all the way through. If it's high first, it stays high. And that suggests that maybe something is clamping the whole system at the very beginning when it's expressed. It's not over a long period of time. So we're very curious about sort of a critical period for this happening and trying to understand how that takes place. So there's too many questions, which is sort of fun.
Yeah.
Ronald.
It is a very interesting and excellent talk. I have two questions, maybe they are related. The first question is, even in the presence of the Guarantor transcription factor, we still see a Gaussian distribution, although over a narrow range. So why is that? What is your thought on that? That is the first question. The second question is, would you like to explain the penetrance of a mutation in this light? Penetrance of a mutation in this light.
Okay, so the first thing is that all the other, I imagine, that all the other biological events are taking place and that there is going to be some variability. That even if something is locked in place, it may have a particular type of probability. Other, who knows what other molecules bump into it or so. So you're going to get some sort of distribution. The incredible thing here is how, in both cases, that distribution has been narrowed. So it's a little bit looking at a glass half empty and half full, right? I think you're looking at, well, it's still distributed, and I'm looking at, wow, it doesn't have this junk here. So I think there's going to be some variability, but what these have done is narrowed the variability such that the output is ensured. The other question was, does this explain
Penetrance.
Penetrance. So for those of you that aren't geneticists, we often talk about a phenotype as being is changing by expressivity and penetrance. And to give you a very gross example, if you had something that was needed to make fingers, a completely penetrant would have 5 fingers on everybody's hand, but a mutant that didn't have this might it might be missing 1 finger or 3 fingers or some other set of it, but there would be a missing finger all the time. Expressivity is how many fingers are left. So that's the variability but the penetrance is how many of the animals with the genotype or organisms with the genotype have the phenotype, and 100%, so variable penetrance. So this is sort of an expressivity change rather than a penetrance change, but after all, they sort of run together because eventually, you might have no effect on some and then a graded effect all the way through. What we were really surprised about was the extent of the expressivity, the change, the variability in the expressivity. That's what gave us our first clue that this might be something interesting to look at.
So do you think that alternative splicing may have a role in the functioning of these guarantor, like whatever guarantor proteins or alternative splicing? Because sometimes maybe it can be speculated that if one guarantor may not work, or like the cell produces another one. So like, what do you think?
So you're raising an interesting point about whether guarantors are actually
Yes,
redundant as well.
Are they actually like?
And the answer is I don't know. I tend not to think that they are.
So because that opens up a new dimension now into the understanding of like.
Well, to me it would cause a problem. At least for us, the whole idea here is that it's a single component system that does it. But in other cases, it may very well be that situation. And certainly in vertebrates, this would be something to really consider, because vertebrates have this quadruplication of the genome. So instead of having one set of Hox genes, they have 4 sets of the Hox genes. So for many of the Hox genes, there's actually 4 different genes that encode the same thing. And so they are absolutely known to be redundant. Mario Capecchi at Utah did experiments on mice in which he eliminated these genes, and you look at gene expression, what's made from a given gene, it's all over the place. But you get rid of it, only a few cells are affected. And that's because the redundancy, there were other cells that did it. So there could very well be redundancy. To me, that just adds another complication.
Okay, thank you.
The one in the second last row.
Yeah, that was an excellent talk, Professor. And I work on C. elegans, and I thoroughly enjoyed your talk. So my question is, although one could expect the basic touch response would be the same for both the sexes, I was wondering, did you check any sex-specific responses, like whether you have a different response for male compared to the hermaphrodite?
So we did not do that. I'm sure we looked at it. Shagu did look at it to some extent, but not extensively. So it could be, but these cells don't have any sexual dimorphism to them. And the Hox proteins are expressed basically the same way in both sexes. So at the moment, that's a lower priority, but I have a vague memory that we actually did look at that, but not sure.
The one this side.
So there is 6 neuron cells. If the mRNA levels of these 6 cells are homogeneous, or what is the quantitative measurement? It is some cells have the higher mRNA levels as compared to other one. If you conditionally remove some specific mRNA, for example ALR, it will transfer for example 6 cells is there. If you remove this mRNA 3 cells, what will be the fate? It will transfer the mRNAs already expressing in the 3 cells to other cells?
Okay, so I'm not sure I entirely understand your question, but I'll try to answer what I think I think I have it. So what you're asking is, what if 3 of the cells were gone?
Yeah.
Would there be other cells that would take their place? Sort of a developmental regulation. And although some things like that happen in C. elegans, it does not happen in the C. elegans nervous system. So there's a particular cell called the anchor cell. 2 cells are made. Either one could be the anchor cell. If one is killed, the other one becomes the anchor cell. It's a skin cell. So there is that sort of developmental replacement, but not in this system. If we kill these cells, there's nothing that replaces them. If we kill the precursor to the cell, there's nothing that some other cell then comes to differentiate like that. In addition, if you were saying, do any of the cells release something that actually affects others, they do have neurohormones in them, but that not to become another touch-sensing cell.
Okay, thank you.
Yeah.
Okay.
There's a woman right there.
Where is that? Yes.
Thank you, sir, for the talk. I have a very small question. Sir, you said the system is very robust in terms of because of the redundancy. So how do you think that in spite of so much redundancy, the cell maintains a very delicate balance of specificity as well?
I'm sorry. Could you just ask the question again? How does the cell become the specific cell if there's so much redundancy?
No, sir.
No.
I'm talking about transcription factors transcribing.
Yeah.
So not all transcription factors can transcribe every gene. So
Yes.
how do they, like, in spite of the redundancy, how do they maintain the specificity?
So I think those are two if I understand you correctly, I think they're two independent events because the specificity has to do with binding and not whether there's another molecule there, right? So one could imagine that if you have redundant transcription factors, one will bind or the other will bind, that it shouldn't make any difference which one, and then it'll turn it on if they're truly redundant. One could imagine if you have too many, there might be some sort of competition and concentration might get too high, so there's a limit to how much you can actually make. But I think these are two different things. One is binding, and the other is the fact that there's more than one component that can bind. So I don't think they interfere, it doesn't interfere with the specificity.
Thank you, sir.
Professor Raghavendra.
I can't hear you, I'm sorry.
Plants are also be quite touch sensitive, like Mimosa purica. I think we have
Mimosa.
Mimosa.
Yeah.
And then what I understand is when you touch some of these touch sensitive ion channels, they push ions out so that the just leaf droops down. But do you have any comments on this touch sensors?
I know that people like Elizabeth Haswell have been looking for years at the mechanosensing components in plants. It has proven to be a very difficult area, but it's not one that I'm very knowledgeable in, so I can't comment about these things. Why tendrils tend to come and wrap themselves around things, and when they hit a post, they start moving around them. Why mimosa plants, Venus flytraps, the things that have overt movement when you touch them, that how those things work. I could probably learn much more from you guys than I would be able to say up here. At the moment, the current thinking about eukaryotic animal touch receptors has come from experiments that we've done, but way before we did it. What's his name? I'm blanking on the guy's name. It's been done with the hair cells of the inner ear. It's been done with fruit flies, in which people have looked at mechanically gated systems and compared them basically with the visual system. So in the visual system, there is a delay of from the stimulus of light, there's a delay of several milliseconds. I think it's 7 or so milliseconds before you get a response. And so this and what people know about how these systems work has led to the idea that there's a chemical intermediate that has to build up or change at this time. But for mechanically gated systems, they work in less than a half a millisecond, and that suggests that there's no possible way for a chemical intermediate. And this works in these cells that we've studied electrophysiologically, bristles that are mechanosensors in Drosophila and in the hair cells in vertebrates. And so that has led to the thought that the mechanosensing component is actually a channel that's directly opened or closed so that it's the change in ions which can occur much faster than a millisecond. Usually, hair cells, I think, is like 10 nanoseconds, or microseconds rather, that are affected. So the common thought is that these are channels that are forced open by the mechanical perturbation in bacteria. Ching Kung's lab at Wisconsin discovered components that were channel proteins in the membrane that as the coli swelled, because it was put in a hypotonic solution, as it swelled, that immediately, it got to a point where it immediately opened channels on the surface, and this the force in the membrane as it gets expanding opened the channels and let solutes out of the cell. And as a result, the pressure went down. So that mechanically gated system, again, looks like a channel. So we have several now examples and all the best candidates for the actual transducing molecule, including the one that we have in our system, are in fact channel proteins that are gated in some way by mechanical perturbations. How that actually is taking place is often very hard because we're still just beginning to get the X-ray crystallographic structure of some of these proteins. Yeah. Yeah.
Okay. Please take a seat. On behalf of University of Hyderabad and on behalf of everybody here and also on behalf of those who have been watching live stream and on behalf of the IRDAI, I have the pleasure of honoring our guest, our speaker, Professor Martin Chalfie, with a small memento and a tradition of a shawl.
Let's see what happens.
This is the memento.
Ah, thank you. Great.
Thank you so much.
Thank you.
I request Dr. Srinivasulu to present vote of thanks.
Thank you, sir. It's my privilege to propose vote of thanks on this occasion. On behalf of the organizing committee, I extend my sincere gratitude to Professor Martin Chalfie for visiting University of Hyderabad and delivering a talk in Hyderabad lecture as a part of Hyderabad Lecture Series. And would like to thank Professor Balasubramaniam for extending his help in Professor Martin Chalfie's visit to the University of Hyderabad. We are grateful to Insurance Regulatory and Development Authority of India, IRDAI, for sponsoring Nobel Lecture Professor Martin's visit as part of Hyderabad Lecture Series 2018. I am sure it will promote scientific temperament in young generation. We also would like to extend our thanks to Honorable Vice Chancellor Prof. Apparao Podele for his constant support and encouragement in organizing this event. Further, we are grateful to Dean School of Life Sciences Professor M.N.D. Prasad and then Professor Ramaya Garu in extending their help in organizing this event. We wish to thank Public Relations Officer, PR Office and staff for their logistics support and in publicizing this event. I take this opportunity to extend our gratitude to the distinguished guests from Indian National Science Academy, delegates from IRDAI and other participants and various institutions in Hyderabad. Thanks to the system administrator and office staff for their help in arranging facilities in the SLS auditorium. And we appreciate all student volunteers for their enthusiasm and their help in organizing this event. Lastly, I thank all the members of the organizing committee and all faculty colleagues who have been involved in this particular exercise. And of course, my thanks to the print and then electronic media for their efforts to cover this memorable scientific venture. And thank you all for your earnest participation.